Welcome to another edition of CCSD Living Environment. Today we're going to talk about chemical compounds of life. The first thing we need to discuss is the idea that every chemical compound is either classified as organic or inorganic. Every living thing, no matter what it is, every living thing contains both organic and inorganic compounds. So the first ones that we're going to look at are inorganic compounds. Inorganic compounds are everything that aren't organic. So that's a very simple definition, but what classifies something as inorganic? They do not contain both carbon and hydrogen. The key there is both. For example, water, H2O, it has the hydrogen, not the carbon. Carbon dioxide, CO2, has the carbon, not the hydrogen. Table salt, what you put on your french fries, sodium chloride, it doesn't have either one. Obviously this is organic. The problem with these is that they have one, not the other. Remember the important thing, it needs to have both. So the most important inorganic compound, and the one that we need to spend a minute with, is water. Water, or H2O, is the most important inorganic compound found in living things. Living things need water because that's where all of the biological processes take place. Respiration, photosynthesis, any other process that takes place inside of a living cell needs to happen in water. So water then becomes a very important compound to living organisms. There's a couple properties to water that we need to look at. The first one being cohesion, or the tendency for like molecules to be attracted to each other. If you've taken a penny and you've dropped water on it, you notice it forms a dome. Or if you have a glass and you go to pour like soda into it and it gets that dome on top and you slurp it out before it spills on the tablecloth, that property or that idea that allows the liquid to do that is this concept behind cohesion. The next one is adhesion. Adhesion is the tendency for unlike molecules to be attracted to each other. Well, water does this one too. If you remember in the beginning of the year when we talked about measuring with a graduated cylinder, measuring volume, you know that you need to measure from the bottom of the meniscus. And that's because those water molecules pull themselves or like climb up the side of that cylinder. Well, what's really happening is that there's an attraction between the plastic or glass and the water, and that property is called adhesion. So how do these things apply to a living system? Well, the idea comes into play with capillary action. Capillary action is the tendency of water to move upward in a tube of narrow diameter. Like, for example, if you've ever done it with a straw, where you take it and you put your finger over it and you lift it out of the glass and the water's still in there? Well, have you ever taken your finger away? And when you take your finger away, some of that water will stay in the straw. Now, you would think gravity would pull it out, but some of it stays in there. And the reason for that is because the water is attracted to itself, cohesion, and the straw, adhesion. But again, how does that apply to living systems? We don't have straws inside of us. Water moving up from the roots to the leaves in a plant is a great example of capillary action. It's a tube with a narrow diameter, and those water molecules are attracted to each other, and they're attracted to the outside of the tube. So they can actually, or it can actually get pulled up through the plant. And that's how plants get water from their roots to their leaves, even in the super tall plants. So that takes us to organic compounds. Now, if an inorganic compound does not have both, then an organic compound contains both carbon and hydrogen. Organic compounds are larger and more complex than inorganic compounds because remember, carbon wants to form 4 bonds. So when carbon does that, when you put a group of carbon atoms together, it becomes a very large molecule because each carbon atom wants to form 4 bonds. So organic compounds are relatively large. I did not draw all of the pictures on the boards. You have them all in your note packet. So as you're going through and listening to this, please make sure that you refer to those pictures so you can see what some of these compounds look like so you might be able to recognize them should you need to. The first thing that we need to look at is this idea behind polymerization. Polymerization, we're making large molecules or polymers from many smaller molecules or monomers. And there's a place there for you to define polymer and monomer. So what you're doing is you're taking a smaller building block, you're taking a smaller unit, and you're putting many of them together to produce a polymer or many object, okay? Something that's large. Polymers are very large objects. So the monomers are the building blocks for the polymers. There's a couple processes that we talked about earlier in the year. The first one being dehydration synthesis. If you remember with dehydration synthesis, synthesis means to make or build. So it's the process of synthesizing polymers by removing water. Remember, water was what we got out. So if I start with glucose and glucose, I would end up with maltose and water. Water is always a product in dehydration synthesis. So this idea of putting smaller things together to create a larger compound, that's the idea behind dehydration synthesis. Well, if you've got one side of that, you have to have the other. What do I mean? If here we're talking about making, then we need to talk about breaking or hydrolysis. With hydrolysis, it's a process of breaking polymers by adding water. So let's do the reverse reaction. Start with maltose and water, and what do I get out? Glucose and glucose. So in this case, water is the reactant. It's what I'm putting in, and that's what's going to break apart those two glucose molecules. And that's what's going to be our product. So the reactants here are the products here, and vice versa. So you have two reverse reactions that are happening with polymerization. If the monomer builds up the polymer, then when you break down the polymer, what do you get out? You get out the monomer. So the first organic compound that we're going to look at are carbohydrates. You're familiar with carbohydrates. Those are your starches. Those are your pastas, your breads, your cereals, all of that stuff. We're familiar with these organic compounds because of our food pyramid. But we need to look a little bit deeper into some of the chemistry of those compounds. Carbohydrates are the source for most of the energy in our body. They contain carbon, hydrogen, and oxygen. And there's a really important thing that we need to point out here. The hydrogen to oxygen ratio is 2:1. That's true in a carbohydrate, not in anything else. Generally speaking, sugar names end in -ose. And you're saying, well, wait a minute, I thought we were talking about a carbohydrate. Well, carbohydrates are broken into two categories. You have sugars and you have starches. The first that we're going to talk about is a monosaccharide. Remember, mono means one or single. So a monosaccharide is a simple or single sugar. These are the easiest chemicals for our cells to break down, for any cell to break down for that matter. Examples would be glucose, fructose, ribose. Again, sugar names end in -ose most of the time. If I asked you the chemical formula for water, you would tell me H2O. If I asked you the chemical formula for carbon dioxide, you would tell me CO2. You know those. There's another one that I need you to add to that collection that you have already in your head, and that's the chemical formula for glucose. And the chemical formula for glucose is C6H12O6. And I want you to look at that hydrogen to oxygen ratio, 12 to 6, which reduced is 2:1. So for any carbohydrate, if you can see the chemical formula, you're always going to see that the hydrogen to oxygen ratio is 2:1. So if I take two monosaccharides, two single sugars, and put them together, I get a double sugar, or two sugars. We call those disaccharides. So that would be the double or two. It is a polymer of two monosaccharides. An example there would be sucrose, maltose, and lactose. Sucrose being table sugar, which you put on your Cheerios. Maltose you find in seeds. And lactose we know is the sugar you find in milk. Again, these sugar names end in "ose." And finally, if we took many monosaccharides and hooked them together, we would form a polysaccharide, many sugars. So it's a polymer of many sugars bonded together. There's a couple different ones that we need to make sure that you're familiar with. The first one being starch. Starch is actually how plants store their glucose. The next being glycogen. Glycogen is how we as an animal store our glucose. And then the last one I want to mention is cellulose. Cellulose is also a polysaccharide or a starch. Cellulose is what forms the plant cell walls. We can't digest cellulose. We don't have the ability in our digestive system to break down cellulose. So then why should we eat it? Because this is the source of fiber that you hear about that's so important for people to eat. And what that fiber does is it kind of helps to clean out your large intestine. I'm not saying that the fiber will prevent you from getting colon cancer. What I will tell you, though, is that by having that cellulose in there, by having that fiber in there, it helps to clean things out. So maybe it will reduce the chance that you might get something like colon cancer. And this is why it's so important that you eat fiber or that you eat your fruits and vegetables. And that's why they say an apple a day keeps the doctor away. It's because of that cellulose. It helps to kind of clean out the system so that things can't build up and cause problems later on. The next organic compound we're going to look at, lipids. This is why food tastes good. Lipids are the answer. Lipids are your fats. So lipids are commonly known as fats, oils, and waxes. If you think about all of the stuff that we really love to eat that we shouldn't, like French fries and potato chips and candy and all of that stuff, that's where our fats are. So yes, the fats are there. They function as long-term energy storage. That's a good thing. They cushion our body organs. They provide insulation. Those are good things, but the problem is we get too much fat in our system and we can't get rid of it. We can't break it down. We can't use it all. That's the problem. A lipid, or a fat, is a polymer, again, a large compound, made of the monomers, fatty acids, and glycerol. So if I'm going to produce a lipid, if I'm going to produce a fat, I use 3 fatty acids and a glycerol molecule, and that gives me a lipid. Well, we need to look at lipids for a minute. Fats are mainly found in animals. They're solid at room temperature, like butter. Those are your bad fats, what we call saturated fats. So what I did is I drew a fatty acid chain for you. It's just a long chain of carbons. And what you'll notice is that surrounding every carbon is hydrogens. This chain is completely surrounded or saturated with hydrogen atoms. Let's take bacon, for example. We all like bacon. When you cook bacon, the fat from the bacon becomes a liquid, and you drain it off. If you let that liquid sit on top of your stove, within a relatively short amount of time, you get that kind of white, slimy fat stuff. It becomes solid at room temperature. That's a saturated fat. It came from an animal. Those are the bad fats. Those are the things you don't want to eat a lot of. I'm not saying don't eat any. We don't want to take bacon away from anybody. But you don't want to eat a lot of them because it's just too much. It's too much of that bad fat, that saturated fat. So what's the other option? Oils and waxes. Oils and waxes are mainly found in plants. They're liquid at room temperature. If you will, these are the good fats. These are your unsaturated fats. Well, what's the difference? Again, look at that fatty acid chain. I drew that long chain of carbons out, and what you'll notice is that surrounding the carbons, they're not completely surrounded by hydrogen. Some of the hydrogens aren't there. And that's because there's a double bond between some of the carbons. And when you put in those double bonds, it's now unsaturated. It's not completely surrounded by hydrogen. That makes them liquid at room temperature. Olive oil, peanut oil, sunflower oil, safflower oil, wherever you get the oil from, These are much better fats for you to consume than the ones from animal fats. That's why it's better to cook in olive oil than it is to cook in butter. So that's one of those transitions you should make, and this is chemically why they're different. These guys are definitely better for us than these guys are. That takes us to proteins, or you'll notice polypeptides. Again, poly being many, because a protein is a polymer. Proteins are probably the most important one we're going to do. Proteins are basically what we're completely made up of. We're a big walking bag of protein. And we'll talk a little bit more about how those proteins are made in our next unit. But for now, proteins or polypeptides, we can distinguish a protein because of the presence of nitrogen. A protein has nitrogen in it. As opposed to carbohydrate or lipid, notice there's no N. There's no nitrogen. So yes, all of them have carbon, hydrogen, oxygen, but protein has the nitrogen in it. The other ones don't. They're made of long chains of amino acids, so the amino acid is my monomer, and they're bonded together by something called a peptide bond. Remember we talked about carbon atoms and carbon wanting to form those covalent bonds, those strong bonds. Well, a peptide bond is just a type of covalent bond. It's a strong bond that forms between amino acids. If I put together two amino acids, I will form a dipeptide. Di being two, peptide for the bond. So a polypeptide is many peptide bonds, and that's what we call a protein. So proteins are made up of amino acids. There are 20 different amino acids. There's 20 of them out there. That's a fact that you should know. What I did here is I drew for you what an amino acid would look like. So you've seen what carbon dioxide looks like. If you remember the other day, I drew methane on the board and ammonia. We've looked at these molecules. This is what an amino acid would look like. There's a couple things that are important to amino acids. The first one is this nitrogen group, or NH2. That's your amine group. That's the amine group for the amino acid. Then you have this C double bond O, OH. This is your carboxyl group. The carboxyl group is what makes it an acid. So when you put the amine and the acid group together, you make an amino acid. I know it seems very trivial, but it becomes important if you can understand how that works. The last thing we should look at, and I tried to circle it up here for you, is this R group. The R group is what they call the variable group. The R group changes from one amino acid to the next. That's how we can tell one amino acid from another. So the R group, how many are there? 20. That's right. There's 20 different amino acids, so there's 20 different R groups. And every amino acid has its own R group. That's how we can tell one from the next. So with these amino acids, when I hook them together, that's what gives me a polypeptide or a protein. The most important thing for you to remember about a protein is its shape. And that seems kind of weird because there isn't a lot of shape there. But remember, this is an amino acid. We're hooking a whole bunch of amino acids together, and that's what's going to make our protein. So why does the shape matter? Well, I'll show you. Shape matters because we need to talk about the structure. Proteins are hormones, antibodies, pigments, and enzymes. And if you remember, just a minute ago I said we're like a big walking bag of protein. Well, what did I mean by that? We have all kinds of hormones. Adrenaline, testosterone, estrogen. There's tons of hormones that make us up. Antibodies are those things that our immune system produces to fight off infection or disease. Pigments are what give our hair and eyes and skin some type of coloring. And enzymes are the things in our cells that run all of those reactions that need to occur for us to survive. All of those things are proteins. That's why the most important thing to remember about a protein is its shape. But where does the shape or the structure of the protein come from? The shape of the protein is determined by that sequence of amino acids. So there's 20 different ones that we can hook together, and depending on the order that you hook those amino acids together with, that will make a different protein. And if you change the order of a couple amino acids, you'll get a completely different proteins. Proteins are such an important part of biochemistry, and we'll talk more about how proteins are actually made in our next unit. And so the last thing that I want to look at, the last and final organic compound, is a nucleic acid. Nucleic acids are made of something called nucleotides. So nucleotides are the monomer, nucleic acids are the polymer. They're made of nucleotides and they contain the hereditary information. Hopefully if I say hereditary information, you automatically think about DNA. Deoxyribonucleic acid, or DNA. It is the hereditary units within the organism. They serve as the blueprint for RNA, and generally speaking, they're double-stranded. It looks like a spiral staircase. This structure here is in your note packet. This is a nucleotide for DNA. The difference being here that the sugar in DNA is deoxyribose, sugar ends in ose, and the bases for DNA could be A, C, G, or T. The other action is a very similar cousin to DNA, if you will, called RNA, or ribonucleic acid. RNA directs the making of proteins. That's interesting. And it's generally single-stranded. Doesn't look like a spiral staircase. It'd be like if you took a ladder and you cut off one side. That would be what RNA would look like, where DNA is the whole ladder. Okay? Again, the nucleotide looks very similar, very much the same, except the sugar here is ribose, ose for the sugar, ribose, and in RNA the bases are A, C, G, or U. And we'll get into more about nucleic acids and nucleotides in the chapter to follow when we start looking at modern genetics and we start looking at some of the information about DNA and RNA. That takes us through chemical compounds of life. Thanks for watching the video, and we'll see you soon.